We will demystify CRISPR gene editing, how a bacterial immune system became a programmable DNA editor, the molecular steps that enable precise edits, notable clinical successes, and the ethical and technical challenges that will shape its future. Origins and fundamentals. From bacterial immunity to a programmable tool, CRISPR was first recognized as an adaptive immune system in bacteria. Clustered regularly interspaced, short palindromic repeats, CRISPR, record short DNA fragments from phages, and CRISPR-associated Cas proteins use those records to recognize invaders. In 2012, Jennifer Doudna and Emmanuel Charpentier showed that a synthetic guide RNA can program Cas9 to cut a chosen DNA sequence. Streptococcus pyogenes Cas9 uses a 20-nucleotide guide paired to a target adjacent to a PAM sequence. SbCas9's PAM is NGG. The human genome contains about 3.2 billion base pairs, but a properly designed 20NT guide can target a unique locus, enabling highly localized genome edits when delivery and cellular repair pathways cooperate. How CRISPR-Cas9 and new editors actually work. Mechanistically, a guide RNA, approximately 20 nucleotides, directs Cas9 to a complementary DNA sequence next to a PAM. SbCas9 makes a double-strand break. Cellular repair then follows two main routes. Non-homologous end joining, NHEJ, is error-prone and produces small insertions deletions, indels, used for gene disruption. Homology-directed repair, HDR, can install precise sequences but is inefficient. HDR rates are often under 10% in many non-dividing somatic cells. To avoid double-strand breaks, researchers developed base editors, C to T A to G conversions, and prime editors, 2019, which use a nickase Cas fused to reverse transcriptase to write specific edits. Delivery AAV vectors, approximately 4.7 kb capacity. Lipid nanoparticles, or ex vivo electroporation, strongly influences efficiency and safety. Applications: clinical trials, agriculture, and research tools. CRISPR's versatility has produced tangible applications. Ex vivo therapies edit hematopoietic stem cells to treat sickle cell disease and beta thalassemia by disrupting regulatory elements of BCL11A, raising fetal hemoglobin. Clinical reports show restored hemoglobin function and transfusion independence in treated patients, with high allelic editing in many trials. In vivo approaches have shown promise. An early lipid nanoparticle therapy for transthyretin amyloidosis, NTLA 2001, achieved approximately 87% mean reduction in serum TTR in a dose cohort. Agriculture uses include mushrooms with reduced browning and crops with improved stress tolerance. In research, pooled CRISPR screens can interrogate libraries of thousands to tens of thousands of guides to map gene function at scale. Dozens of clinical trials are ongoing for a range of indications. Advanced techniques, risks, and ethical implications. Newer engineering reduces risks and expands scope. High-fidelity Cas9 variants, lower off-target cuts. Base and prime editors minimize double-strand breaks, and epigenome editors modulate gene expression without changing sequence. However, challenges remain. Off-target edits, mosaicism, immune responses to Cas proteins, and delivery to specific tissues. The 2018 germline editing case, He Jiankui, triggered international condemnation and legal consequences. He was sentenced in 2019, leading to calls for moratoria on human germline edits. Economic and access issues matter too. Current ex vivo therapies can cost hundreds of thousands to over $1 million. Regulatory frameworks, public engagement, and equitable access will determine responsible deployment. CRISPR transformed a bacterial defense into a flexible genomic toolkit. Programmable targeting via guide RNAs, multiple editing modalities, nuclease, base, prime, and accelerating clinical impact. Technical refinements and robust ethical governance are essential to realize CRISPR's potential safely and equitably across medicine and agriculture.